Hi everyone, my name is Sebastian, and today I'm going to be delving into a paper that basically goes into how our personal gut physiology has a direct impact on our human gut microbiome. So, why should we care? Well, the human gut microbiome has been found to heavily influence a multitude of health conditions in the body, from gastrointestinal disorders such as colorectal cancer to even metabolic diseases. And although diet is indeed a pivotal factor that shapes our microbiome, it does not account for all of the variability seen in our gut microbiota, which is why the authors of the following study asked the question: how might personal gut physiology and environment influence the differences that we observe in the human gut microbiome? Well, he set about answering this question by executing a 9-day observational trial of 61 healthy adults using what they called multi-omics to profile their microbiomes and metabolisms. And then they observed daily changes and measured transit time and pH using a wireless motility capsule called a smart pill in a 50-person subset, concluding that both transit time and pH largely determine the composition and metabolism of the gut microbiome. So, for some background, prior to this study, research had already existed on the role of diet in the gut microbiome in particular. For instance, high-fat diets were looked at with concern as they were found being capable of leading to microbiota dysbiosis. On the other hand, prebiotic, probiotic, and fiber-rich whole plant-based foods have been found to be excellent dietary choices to maintain a healthy gut microbiome that is efficient and free of any uncomfortable or unfavorable symptoms. Although the role of diet was still strongly valued as a core principle for gut health and microbiome health, it was still also acknowledged that other factors such as genetics, medications that people could be taking, or even birth delivery method could influence the composition of that person's own microbiome, and their gut microbiome in particular as well. So why the focus on this particular question? Well, as you can see off pictured to our right, is a diagram of the large intestine actually, and it's by the National Institute of Diabetes and Digestive and Kidney Diseases from the National Institutes of Health. Statistics actually from the American Cancer Society show that when excluding skin cancers, colorectal cancer is the third most common cancer in men and women in the United States. And even further, in people under 50, every year from 2012 to 2021, the rate of colorectal cancer diagnosis increased by 2.4%, which is pretty major. So, by investigating what internal bodily factors other than diet could predispose someone to microbial traits that can lead to issues with their gut health and even beyond, seeing as though the gut microbiome is kind of the gateway to many other health conditions of health in our body. By investigating what other than diet could be causing these issues, we can improve our understanding of how to prevent and treat these conditions across a much wider spectrum of people if we have this deeper level of understanding and knowledge on the internal goings of our gut. So as far as methodology goes, the authors of the study conducted a 9-day observational PRIMA study. PRIMA meaning, in their words, toward personalized dietary recommendations based on the interaction between diet, microbiome, and abiotic conditions in the gut. So 63 healthy participants at the Department of Nutrition, Exercise, and Sports at the University of Copenhagen in Denmark were enrolled, yet only 61 completed the study. Now, to answer for this, the authors explained that the 2 participants who did not continue with the study were excluded because of illness and antibiotic administration as a result. So the 61-person cohort continued on without them. It consisted of 43 women and 18 men and ran from April to December 2021. And of this study population, a subset of 50 volunteers underwent wireless motility capsule monitoring from Smart Pill at visit 1. Now, there were 2 physical visits over the course of this 9-day trial, and we are referencing the first one now, in which they underwent the monitoring from the Smart Pill capsule. 7 days prior to the beginning of the study, the participants were actually asked to refrain from consuming sweet corn, actually due to the 2 self-administered sweet corn tests that they would perform on themselves to measure their whole gut transit time. And this transit time basically just means, this whole gut transit time basically means the time that it takes for food to pass through your entire digestive system. Also, before the 2 physical visits, participants were asked to refrain from alcohol consumption, smoking, or strenuous exercise, pretty common types of things. Participants also were asked to continue their habitual diet and record their food intake online through the MyFood24 tool for 8 consecutive days, getting their nutritional values from Frida, which is a Danish food composition database. During the study, the participants did collect their daily stool samples, preferably the first bowel movement of the day, but this was not always possible, so whatever they could get daily. They stored these samples in their freezers, their domestic freezers, before they would then be sent and transported to the lab. Participants also self-reported their daily time of defecations, their stool consistency according to the Bristol Stool Scale, which measures stool consistency and basically classifies it. They also reported their stool frequency, as well as their intake of supplements and medications, and any gastrointestinal symptoms that they might have been experiencing. Participants collected 7 daily morning urine samples and 2 24-hour urine samples, which were stored in their freezers before being transported to the study site. And at both physical visits on day 2 and 9, fasting blood and breath samples were collected. Now, on the more technical side of things, 16S ribosomal RNA gene sequencing, something that we've covered a lot, we should be a bit familiar with by now, at least the name, using ion torrent semiconductor sequencing technology was used, as well as Smart Pill data collection and analysis, as I referenced before. So specifically the Smart Pill, it collects data on luminal pH, temperature, and pressure as it's ingested by the participant, as it's a wireless gastrointestinal capsule. And then it sends the data to a receiver that the participants wear. And then that data can then be taken and analyzed. Dietary records were taken for 8 consecutive days through the MyFood24 online tool. Breath exhalation measurements were also performed. Also, biochemical analysis of blood, fecal measurements, microbiome and metabolic profiling, a UPLC electrospray ionization QTOF, MS analysis, metabolomics, data processing using MS Convert, open source R packaged XCMS using the Scentwave algorithm, and which used high performance and ultra high performance LC system that was coupled to a mass spectrometer, and of course statistical analysis. So for our results, we see figure 1 pictured right. So this is figure 1 of our results. Of course, this is a PRIMA study design, as we mentioned. And the first thing right off the bat that was basically found was the fecal pH, Bristol Stool Scale results, stool frequency, stool moisture, and microbial load vary daily. They had very daily fluctuations within individuals. So what this basically meant was that these factors fluctuated within people, within each of the participants daily, and then across the participants, these daily fluctuations were also varied. So different people had different forms of fluctuations daily. Most gut environmental factors, therefore, were noticeably varied. Besides fecal pH, which was much more stable. And so the main takeaway was that the stability of the gut environment varied for each person in the study. As we move on to figure 2 of the results, we see distance-based redundancy analysis was performed, and it actually came to the conclusion that diet did not explain intra-individual fluctuations. Instead, stool moisture, fecal pH, BSS, which is the acronym for the Bristol Stool Scale, and time of defecation, actually all affected the gut microbiome. And therefore, the conclusion is that variations in gut transit time are correlated with variations in the gut microbiome, seeing as though those factors are basically a proxy for the gut transit time. Also in figure 2, notably stool moisture and fecal pH explain some intra-individual variation in urine metabolomes at 3.1% and 3% respectively, which did suggest that colonic water content and pH could be associated with microbiota metabolism. Either that, or alternatively, it could be just due to day-to-day dietary variations. As we move on to figure 3 of the results, this is where we come to the finding that gut environmental factors were not found to influence intra-individual variations in fecal metabolomes. And here we see that smart pills, what we were describing earlier, were used to measure the whole gut transit time, gastric emptying time, small bowel transit time, colonic transit time, and the intestinal transit time, as well as the pH throughout the gastrointestinal tract. And it was found that transit times and pH were found to vary between people. And transit time itself was found to be negatively correlated to Bristol Stool Scale results. Additionally, DBRDA was performed using data from the fecal and 24-hour urine collections that were received from all study participants on day 2. And stool moisture and distal colon pH were associated with inter-individual variation in QMPs. And QMPs basically just stands for quantitative microbiome profiling. And the stool moisture was associated with variation at 5.5%, and a distal colon pH at 5% variation. And the Bristol Stool Scale (BSS) was not found significant. Meanwhile, the whole gut transit time, colonic transit time, colon transit time, and fecal pH explained 9.1%, 6.2%, 4.9%, and 5.4% respectively of inter-individual variations in the 24-hour urine metabolome. Now the authors go on to note that first, age only explained 4.5% of the variation, and additionally, quote, that segmental transit time did not contribute to the inter-individual variation in the fecal metabolomes, whereas pH in the distal colon and fiber intake showed the largest effects, explaining 6.8% and 5.9% of the variations, respectively. However, this was not significant after adjusting for multiple testing, so that was an important point made there on the author's part. But the authors did maintain that personal gut microbiome did contribute significantly to inter-individual differences in the gut microbiota and urine metabolomes. Inter-individual fluctuations in microbial-derived metabolites, including breath hydrogen and methane, fecal short-chain fatty acids such as acetate, propionate, butyrate, valerate, and caparate, and BCFAs like isobutyrate, isovalerate, and 2-methylbutyrate were measured. Also, 16 other microbial-derived metabolites that were detected in feces and urine, such as proteolytic markers, PCH, which is p-cresyl sulfate, and phenylacetylglutamine and indoxyl sulfate. Those were also measured. The actual data came out to suggest that microbial-derived metabolites that were in the breath, feces, and urine fluctuated daily in a person on a habitual diet. So even eating the same kinds of foods basically daily, these metabolites fluctuated in spite of that. Also, colonic transit time and colonic pH were found to be associated with levels of microbial metabolites in the breath, feces, and urine. Meanwhile, small bowel transit time and small intestinal pH were actually not. As we move on to figure 4, we have it pictured on the right, and untargeted metabolomics were used specifically here as a methodology to profile the urine and fecal metabolomes. Urine and fecal metabolomes were found to be linked to whole gut and segmental transit time, as well as pH as a main finding. And specifically, the urinary metabolites 5-hydroxy-2-oxindyl sulfate, 3-hydroxy-2-oxindyl sulfate, and 4-hydroxybenzoic acid sulfate were associated with a longer whole gut transit time and colonic transit time. 3-hydroxy-2-oxindyl glucuronide was, on the other hand, associated with higher fecal pH. And interestingly, the authors actually noted that urinary 4-methylcatechol sulfate, which is a metabolite of quercetin, which is actually found in plant-based foods, was positively correlated with rectal pH. The authors then remarked that the results show multiple host, microbial, and food-derived metabolites are associated with whole gut transit time, colonic transit time, and pH, and the distal part of the colon. And they maintain that this is consistent with the idea of diet, the gut environment, the host, and the microbiota all being significant factors. So this intersectionality between all of these factors kind of contributing to the state of the microbiome. So all of them kind of having some influence and sway. Thus, all of them being significant factors. Figure 5 is pictured right, and here we see the Spearman correlation analysis that was performed to measure association between the metabolites and gut microbiota. Microbial alpha diversity was found to be positively correlated with microbial proteolysis, colonic transit time, and intestinal transit time. Microbial alpha diversity was found to be negatively correlated with stool moisture and microbial saccharolysis, on the other hand. And microbial saccharolysis, well, that's basically just microbes breaking down sugars. And these sugars basically are carbohydrates, and they're doing that basically just for energy. So that's basically microbial saccharolysis. Now, the authors mentioned that, quote, products of microbial proteolysis and dicarboxylic acids were positively correlated with the absolute abundances of several bacterial genera, including Intestimonas, Flavinifractor, Eubacterium, Lactospira, Clostridium, Oscillobacter, Alistipes, Dialysir, and Akkermansia. These genera were found to be negatively correlated with fecal levels of tryptophan, oxygen-3-acetic acid, and coffee-derived metabolites. Additional positive association was found with longer intestinal transit time and colonic transit time and higher fecal pH, while negative association was found with stool moisture and or Bristol stool scale results. Also, worthy of mentioning are the short chain fatty acid producing genera Acinetobacter, Faecalibacterium, and Bladia, as well as the lactate producing Streptococcus, which were all found to be positively correlated with fecal nicotinic acid, pantothenic acid, and coffee derived metabolites, yet again. The authors note that Ocelobacter, Allostypes, and Akkermansia were linked to longer transit time and or constipation. And in direct contrast of this, butyrate producing genera like Faecalibacterium and Acinetobacter were on the opposite end of the spectrum and were linked to shorter transit time. And this is kind of an important distinction, and I think an important time to mention that neither extreme is very good with transit times, which again is the time it takes for the food to pass through your digestive system, especially with longer transit times. It kind of lead to the constipation-like symptoms, which have kind of been which have been historically, I should say, correlated with some of the symptoms one might experience from various gastrointestinal disorders, like even colorectal diseases, like we were discussing earlier. So, as shown, the composition of the human gut microbiota can be quite sensitive to a multitude of factors within the gut itself, particularly as the authors definitely highlighted throughout the entire study, particularly transit time and pH. And because we now know of the importance the microbiome holds in our health, including in the colon, it is reasonable to explore what may internally cause it to be out of whack or to fluctuate, so to speak. From these results, we can gather that modified transit time can swing the gut microbiota in either a state of saccharolytic or proteolytic fermentation. So, on the one hand, it could be saccharolytic, meaning shorter transit times, or proteolytic, longer transit times. And overall, the amount of variation in transit time and pH seen between individuals was, in my opinion, quite interesting, and notably contributed to the microbiome's compositional and metabolic differences. And not to mention, based on the importance of transit time and knowing that microbes that produce butyrate are linked with a shorter transit time, I did also think it would seem reasonable to promote this short chain fatty acid, being butyrate, through dietary means. And I think once again this is where we see the multiple factors coming into play, that both diet and physiology, and of course other factors, your environment and you know medications or whatever it may be, where they're all kind of intertwining here, and there's that intersectionality where the factors could maybe work against you, or they could possibly even work all together to benefit you and your gut microbiome and the microbial communities there. So, for example, as maintained earlier, consuming foods rich in fiber that can promote butyrate production in the gut through fiber fermentation should be a healthy action that actually improves the overall health of the microbiome, while correlating with a shorter transit time that is kind of the opposite of the longer transit times that were mentioned earlier. So, to conclude, I just think it's fascinating to think about how our guts are unique, just like us as people. And in all, it seems that going forward, both diet and individual gut physiology should be considered when performing treatments, especially for people with unhealthy microbiomes or other similar gut issues, disorders, who may even be at risk for or battling certain types of colorectal cancers. And I just hope that this does lead to more research in the field, specifically on um, kind of how to improve our diet to work with our internal gut physiology, and also looking at the gut physiology and what we can do to improve that, and just more research into how exactly, even over probably wider populations, how that seems to affect our microbiomes as humans as a whole. Um, and then I have some questions for the group discussion. So 5 here. First, just do you think altering transit time, do you think that could be as impactful as probiotics or dietary change for shaping the microbiome? Also, do these physiological factors confound microbiome-based diagnostics? How might medications that alter gut motility, like laxatives and opioids, affect then sorry, microbiome-mediated health outcomes? Um, are slow transit individuals more at risk for proteolysis-derived toxins? And what study design do you think would be needed to test causality um, rather than just association or correlation, as was done in this study um, for RCT altering transit time or pH, for example? All right, and this is my list of references. The references I used in addition to, of course, the article that was my focus of this review for Journal Club. Thank you, everyone, for listening.